only thing we're doing today, bacterial transformation, right? And we're using a particular system put together by Biorad, and so usually always works. I was burning my hands last night making media for you guys, so it should be all good. Tried to take it out of the microwave before it was cool. Not a good idea. So this type of technology, right, this use can be done even for eukaryotes. So these mice, you can see 2 of them are glowing out of the 3, right? And one of the main reasons why scientists use these proteins is because of the ability to just shine a UV light on them and you can see them glow. Transformation doesn't always happen, right? We don't always get them to take up the DNA and express the DNA. So this is a nice quick way of knowing whether they took up the DNA and are expressing it, because they're actually glowing. So for this guy in the middle, unfortunately, what they were probably trying to do did not work. So they wouldn't be able to include that mouse in its study. So this is one way of using these fluorescent proteins is very beneficial, right? So other than making pretty things, there is a scientific reason behind doing this. So I actually wanted to show that first. So this is actually bacteria that have different fluorescent proteins that somebody literally drew with bacteria onto a petri dish, right? And so some of the kids in the STEM program have been drawing too with bacteria on their petri dishes. Not anything this complex. They didn't have as many colors to choose from, but they were having fun with that. But here's a real link to a real scientific experiment that was conducted using different colored fluorescent proteins. And so hopefully it will open up for me. There we go. So in this video, what you see swimming around are sperm. You can really see the green ones, right? But look closely and you can see the red ones. Let me play it again. There's more red ones actually. And these are sperm from 2 different fruit flies. So one fruit fly they genetically engineered so that its sperm would be green. And a different fruit fly, they engineered theirs so they would be red. Why did they do that? So that they could look, and this is actually the reproductive tract of a female fruit fly. And they actually mate with more than one male, because they want a diverse population of offspring, so it betters their chances, right, of their offspring surviving. So they purposely mate with multiple males. One of the things that they found, though, especially now that you can tell the difference between one male sperm and the other males, is actually the sperm themselves compete to get to the eggs, right? They try to block each other. And of course, the female, this is not advantageous for her. And they did more scientific testing and found that she actually produces chemicals in our reproductive tract that tries to control this competitive edge of the different sperm, right? But so they were actually able to visualize this, right? Able to tell one sperm from another by genetically engineering them so there were 2 different colors, right? Take it out, film it, and be able to make observations about what's going on. So fluorescent tags, using fluorescence is very common in scientific research nowadays. Being able to either add them to antibodies to stick to things, or in this case, right down to the DNA level, and the organisms are actually expressing that protein. So today, and this happens in the environment. When a cell dies, its DNA becomes part of the environment, and under certain circumstances, other bacteria will pick up that DNA, and they'll incorporate it into their genome. What we're doing today, though, is using a plasmid in the environment and encourage them to pick that up. That's a circular piece of DNA that's not going to incorporate into the chromosome. But most of the time when transformation happens, is they're picking up pieces of DNA and they're incorporating it into their genome. And of course, this gives them a new ability, right? DNA is information. It allows you to do something. So a transformed cell has taken up a portion of the DNA from another organism and has it integrated into their chromosome, right? That's what transformation is. So, how is this important to our daily lives? Well, this movement of genetic information from one bacterial cell to another has led to what we refer to as superbugs. Because remember, it's information. This information could be information that leads to antibiotic resistance. So in December 2009, a Swiss man acquired a Klebsiella pneumoniae infection while in India that was resistant to, and I can't even say this class of drugs, I'll try and not butcher it too bad, carbapenem class of antibiotics. So it's a particular group of antibiotics, right, which it wasn't normally resistant to, but in this case it's resistant. So what happened? So these drugs, similar to penicillin and ampicillin, they have the beta-lactam ring. And so what it is this bacteria picked up an enzyme. And this enzyme was on a circular piece of DNA that we call a plasmid. And this enzyme, because it can break the beta-galactanase ring, it's commonly referred to as BLA. And now that's the genetic information. Beta-lactanase is the enzyme. And so this particular one, because of where it was discovered, right, gets the abbreviation NDM1, standing for New Delhi Melitino. And then, of course, beta-lactanase refers to the actual enzyme. And it's one of many beta-lactanases, unfortunately, out there. So we're going to do the same thing today. We're actually going to give the beta-galactanase gene to our E. coli. They're not normally resistant to ampicillin. We've seen this previously, right? On our plates, it was sensitive to ampicillin. But we're going to make E. coli that are resistant to ampicillin today by transformation. So that movement is what we call horizontal. One bacteria picking up information from another. Vertical is when that single bacteria produces its offspring and passes that information on to them. So it can go from bacteria to bacteria, that's horizontal, and then travel vertically through the population of that specific bacteria. So in August of 2010, 180 isolates, right, of bacteria containing this gene, this BLA, this beta-lactanase enzyme that can destroy antibiotics that have this beta-lactin ring, as well as ones resistant to fluoroquinocones. I always get tongue-tied on this one. Aminoglycosides, one of the ones that we looked at was like, I think, tetracycline. But most were still susceptible to polymyxin antibiotic and colistin. Colistin, however, is toxic to humans, and it affects our kidneys. So this is, of course, difficult to use. So we're running into where we've got bacteria that are resistant to a lot of the drugs that we're using, right? And they've acquired this genetically from other bacteria. Now, how it probably came about was probably due to genetic change in general, and the bacteria got lucky, right, and produced this enzyme. So similarly, metallism-resistant Staphylococcus aureus, right, this one, we believe, again, acquired this enzyme, this ability to destroy from other bacteria. And then the really scary thing is that we have extensively drug-resistant forms of tuberculosis, meaning it's resistant to most of the antibiotics that we use. They have multi-resistant strains, and then the really scary one, right, where I think they're down to like 2 antibiotics that are effective against this tuberculosis out of all the antibiotics we have out there, right? That's really kind of frightening. And then another example of genetic information moving, E. coli 0157. This is a pathogenic strain of E. coli. It has the Shiga-like toxin, and it's believed that it picked up this toxin from a genetic transfer from the Shigella genus of bacteria that have this toxin. And it's also similar to some of the toxins that Salmonella has. And again, a circular piece of DNA right, was transferred from one bacteria to another. It could have been through transformation where it picked up from the environment, or do any of you guys know from lecture class when two bacteria join together by the sex subtilis? What is that process? Conjugation. Conjugation is another way that they could easily transfer plasmids. Another way that genetic information moves throughout the bacterial population is through viral infection, where viruses pick up, and that's referred to as transduction. But we're doing transformation today, so it's just gonna pick up DNA from the environment, right? In this case, we're gonna put the plasmid, the circular piece of DNA, into the environment, basically into the test tube. So, if you're interested in superbugs and the evolution and prevalence, this is a good article. So, cells can become competent, but only about 1% of the known bacteria are naturally competent. In order for them to take up the DNA from the environment, they must be what we call competent. So transformation in nature is a very complex developmental process. So it involves the expression of 40 different genes that are required for the competency, for instance, for Bacillus subtilis, right? It requires energy to take up the DNA from the environment. But it has evolved in a way to repair DNA damage, right? So if they're picking up DNA from the environment, chances are the cells around them were their own cells, right? Own progeny maybe that died because of something that caused the damage. So they could pick up, right, hopefully maybe pieces that were still intact that became part of the environment and repair their DNA. So it's one of the reasons why they believe that this mechanism developed, right? It's one of the ways to repair themselves. So they really only do this in high stress conditions when there's a high cell density or there's nutrient limitation, right? When they're really kind of stressed. It can be viewed as a primitive means of sexual reproduction where they're taking in DNA, because sexual reproduction is really just an exchange of DNA. And then, of course, if they incorporate it, they pass it on to the next generation. We're gonna create artificial competence. One of the ways you can do it is electroporation, which we don't do here in the labs. We don't do a lot of this type of technique. And this, of course, involves high-tech, expensive equipment, right? And using electrodes, right, to basically zap the membranes and make them more permeable. And this will help them take up, so this technology is not only used just for DNA, but to try and infuse chemicals or drugs into cells in different types of research. And this can be done on both eukaryotic cells as well as prokaryotic cells. So remember, bacteria are which? Prokaryotes, right? So we're doing prokaryotes today. So as I said, costly, expensive, you need special equipment, special little vessels to do the electroporation. Another way is chemically treated cells, and that's what we're going to do today. So we're going to use a concentration of calcium chloride, and this is going to produce a lot of calcium ions, positively charged ions, which are going to help make the membranes more permeable. It's thought to bind to the negative components of our phospholipids, thus shielding the cell surface from the negatively charged DNA that's coming in, the exogenous DNA from the outside. And in this case, will help us actually bring in double-stranded DNA, which is what they don't normally do when they do transformation on their own under natural conditions. They take it in single-stranded. So only one strand of DNA is allowed in the cell while the other strand is degraded outside the cell. However, this is enough that you get the homologous, undamaged DNA into the organism and has a slightly damaged chromosome. Excuse me. So commercially available kits, like I said, we get our kit from Bio-Rad. This is one from Inventor Gene. This is used a lot in research, getting the bacterial cells to take up the DNA, and most of the reasons why we use it in research is to get them to copy the DNA for us. And so we can get it to copy huge amounts of DNA that we can later lyse out of the cells and use to sequence DNA. And so one of the things that I did when I was a graduate student was do just this very thing, is take pieces of DNA, insert them into plasmids, and then insert them into bacteria, grew them up, lysed it out, right, and took that DNA and sequenced it. So there's different levels of efficiency, right? How good are the cells at doing this, right? And like I said, ours has a particular efficiency. And of course, the more efficient ones will cost you more money, right? Depends on what you need for your research. So they give you aliquots or competent E. coli. So we're working with special E. coli today that are been made competent, and we're going to use the chemicals so that they will take in the DNA. And so for this particular thing, they also provide a positive control. We will have controls in our experiment as well, and that's just to make sure that the system is working. So what is transformation? It's a process by which bacterial cells take up exogenous DNA molecules, right? So from the outside. It's normally not stuff that's their organisms genome, but it can be. It just depends on where the DNA came from. Competent means having the ability to take up the DNA. And then we're also utilizing bioluminescence today. And this is the production or emission of light by a living organism as the result of a chemical reaction, during which a chemical energy is converted into a light energy. So in this case, it's going to be emission of light. It'll absorb the light and reflect it back to us. It's not actually producing light. But there are some organisms that actually have the ability to produce, from a chemical reaction, light. If anybody watched Shark Week, there are some sharks that bioluminesce. Pretty cool stuff. So in the case for ours, right, the light will be absorbed, and then when the electrons that were displaced are transitioned back, it will fluoresce. In the case of ours, it's going to fluoresce green. But it'll absorb the ultraviolet light spectrum, right? Quite a bit of energy it's going to absorb. So mathematically, this can be calculated. The important thing, so it means that the wavelength, the smaller the wavelength of light, and UV is out of the wavelength of our visible spectrum, right? You can't see UV light. This light, because it's such a small wavelength, it'll emit higher energy, right? And that's one of the main reasons why we use it. So ultraviolet's outside the spectrum, but remember, it's going to lose energy when it's radiated back at us. So we'll actually be able to see the green. It'll be within the visible light spectrum. So all conditions have been worked out already for this experiment, right? We know how many cells to use. We know how much DNA to use. We know the transformation efficiency. So apparently, for this one, we should get approximately 75 transformants for the 100 microliters you'll plate. Normally, you could make a serial dilution and plate a variety of cell concentrations, right, in order to determine the accurate count. We're not going to do that. But we are going to deal with distinct volumes. So the plasmid that we have is a specially designed plasmid called the PGLO plasmid. P for plasmid, GLOW for the fact that the green fluorescent protein will glow. So it has several pieces of genetic information. It has the BLA gene, right? This is for making the beta-galactanase enzyme that will destroy the ampicillin in the plates. So ampicillin won't kill these guys. We have the GFP, which stands for green fluorescent protein. So this is the genetic information, the DNA, to make that protein. That protein, though, we have under a control mechanism, that section of DNA. That control mechanism relies on another protein, which is abbreviated ARAC. This is a structural protein that's going to be produced and bind to the DNA. Where the green fluorescent protein actually originated from was from jellyfish. So they're another thing that bioluminesce. When you go to the aquarium here in New Orleans, that's why some of them are in those dark tanks, so you can see those properties. Although one of them, if you actually read the placard, it's one of my favorite ones, it's not bioluminescing. It has little tiny projections, almost like cilia. And because they're so tiny, when the organism moves, they act as prisms. So it looks like there's a little rainbow running along the edge of them. They're not bioluminescing. They're actually just refracting light, right? Those tiny little projections that they have on them. So they're really kind of cool. So constituent expressed means that it's always made. Right? It's not being repressed or turned on. So we're always, once we give them this plasmid, they're going to make the ARAC, that protein, and they're going to make the BLA, the beta-galactanase enzyme. The GFP they're not going to make. This one's under control. So it takes energy to replicate the plasmid. Many transformants will eventually expel a plasmid sometimes. Were it not for the ampicillin, because the ampicillin puts the selective pressure on them that if they don't have that plasmid, they can't make the enzyme and they can't destroy ampicillin. So this is another thing that will encourage them to keep the plasmid because they can't survive without it in the media that we're growing them in. And so therefore they will become resistant to the antibiotic. So the operating system, and the reason why some genes are under control, and a lot of your genes are under control, they're not always constantly making the proteins from the DNA, is because you don't always need all those proteins all the time. So probably one of my favorite examples, it's actually used a lot in research, although we're using a different sugar, lactose, as it relates to E. coli. E. coli isn't usually in the presence of lactose. So it normally does not produce the enzymes necessary to digest lactose or bring it into the cell. So some people have become what's referred to as lactose intolerant. And what that means is when they ingest milk products, right, because that sugar is found in milk, and so when they ingest that sugar that's found in those foods, when it gets to their small intestine, they no longer, or never did, produce the enzymes themselves to break down lactose. So it doesn't get absorbed in the small intestines. What happens then is it makes it to the large intestines, where you have a very large population of E. coli. They love lactose. But usually they don't see it, right? Because usually you eat it before they get to it, right? Usually you absorb it in your small intestines before they get the chance to eat it. Because they don't normally see it, they don't always have those enzymes ready. But they do have receptors on their surface that can detect the presence of lactose, and right away they'll start making the enzymes. They'll even make more proteins in the membrane to bring in more lactose when it's available. The bad news for us is that E. coli can grow really fast with lactose. Right? When you give it sugar, it's going to grow like crazy. So for some people this will create constipation. Other people, it can create diarrhea. And again, it's because of the overgrowth of the bacteria. And again, the bacteria growing in probably mostly an anaerobic environment, they're actually fermenting that lactose. So it's another byproduct that they produce. Gas. Right? So this can be very uncomfortable for people with this type of intolerance. Usually once they figure it out, they stop eating it. Right? Or they take enzymes when they eat the lactose to help digest the lactose, so they'll actually absorb it. So similar mechanism here. So how does it all work? So operons are a sequence of genetic material that functions in a coordinated manner, right? This is how we're going to control whether we use this genetic information or not. So we have an operator, a promoter, and 2 or more structural genes. So these are the ones that are going to actually make something, right? All this stuff will be utilized at the same time. So what's the job of the operator? The operator is actually a segment of the DNA which actually regulates what happens. So inducers will bind to the regulator and change the way it interacts with the operator. In the case of a ribose operon, that protein that's derived from the gene ARAC is the regulator, while a ribose, the sugar, works as the inducer. So they're going to interact with each other. All right, and I have some diagrams that are going to kind of explain and help us figure out this terminology. The promoter site is a region of the operator, and this is where the RNA polymerase will bind. And what RNA polymerase does is just copies DNA into RNA, right? Which then can be translated by the ribosomes into proteins. Structural genes actually make something, so typically a protein, right? So it's going to make a protein that does something or is structurally important. In this case, they're enzymes that are going to digest. When we talk about beta-galactanase, where the green fluorescent protein, that's it, right? It's going to produce a protein that's going to fluoresce when we shine UV light on it. That's our GFP. So arabinose is the sugar that we're going to use that's already in our media today in one of your plates. It's actually derived from plants, from pectin and hemicellulose. So typically, right, the organism would have enzymes here to digest the sugar arabinose. Here's our ARAC that's binding to the DNA at the operator site. Your effector, or in this case inducer, is arabinose. When that binds, it allows the promoter site to be available for RNA polymerase to attach, and then it's going to read this DNA and make these enzymes. In the case of our system, instead of giving it enzymes to digest arabinose, we just replace those with the green fluorescent protein. So when arabinose is present, it binds to the regulator, right? It allows RNA polymerase to bind to the promoter, and that cell is going to make green fluorescent protein from this DNA. So we tricked it, right? We stole its operating system for a sugar digestion, and we're making them make green fluorescent protein instead. But only when the sugar is present. Because only when the sugar is present will it bind to the regulator and allow RNA polymerase to copy this information. So again, in E. coli's world, it usually does no reason to make the enzymes to digest lactose, because usually it's not going to see it. But when lactose becomes available, the same thing happens. It binds to proteins bound to the DNA, and it's going to cause that cell to actually copy the DNA, those enzymes, to digest lactose. A whole bunch of them all at once. So, green fluorescence only happens if we actually have expression of that gene. Green fluorescence expression leads to green fluorescence, right? It will actually glow. No green fluorescence means no gene expression. Or anybody know another consequence of why you won't get gene expression? If they don't have the gene to begin with, right? But in this case, the pressure we have in this experiment, what's going to make sure they have the gene? They can only grow on these plates if they have that plasmid, right? Because what's going to kill them if they don't have the plasmid? The ampicillin. If they don't make the beta-galactanase enzyme, we'll kill them. So, if part A is true, then B has to be true, right? We can only get green fluorescence if we get gene expression. Flip it the other way. Only when you have expression will you get green fluorescent protein. If you don't have any green fluorescent protein, you don't have any expression. Right? That gene's not being expressed. So can we measure qualitatively and quantitatively? We could measure quantitatively if we count it, right? Quantitative is counting. Qualitatively we can see, does it fluoresce, does it not? Right? So under what experimental conditions do we see fluorescence? Do we observe it? And we should only see it when the gene is being expressed. Right? And then we will support our hypotheses. And inversely, right? If we don't see fluorescence, we shouldn't have expression. So BLA, right, is a genetic information to make beta-galactanase enzyme. This is constitutively expressed. It's going to produce it. And so antibiotics that have that beta-galactan ring, like ampicillin, penicillin, right, all those cillins, this enzyme will destroy that. So before transformation, do they have this genetic information? No. After, they should, right? And we'll be able to know because they won't grow in the presence of ampicillin without this. Okay. So what you're actually going to do today. Steps 1 and 2 in the procedure have been done for you. The tubes are already labeled in your bins, plus and minus. Plus is for the tube we're going to add the DNA to. Minus is for the one we're not. Why are we not going to add it to one of those tubes? What is it going to be in our experiment? Our control. We're going to add already added to those tubes is 250 microliters of transformation solution. This is that calcium chloride solution. So it's already in your labeled tubes. What you're going to do is get the ice buckets, and they're actually Tupperware dishes with ice in them. We're going to add some water, right, and create an ice water bath. That we're going to put those white racks that you have in front of you in. Those will float. You're going to take your tubes that are in your foam containers in your boxes and put them in those racks. So we're going to put those solutions on ice. Then you're going to, from a special plate that I'm going to give you of competent E. coli, you're going to add, and you can use the same loop because at this point both of those solutions are the same. You want to pick up a little bit of bacteria and you're going to put it into those aliquots. Now, Peter said the more likely of getting an accurate was if we put it in one and then split it into the two, but we're not going to do that, okay? Maybe in the future we will. I'm going to show you a solution of the plasmid, which is just DNA, under UV light. So is it going to fluoresce? It should not, because it's DNA, right? What fluoresces? The protein that your cells are going to make from the DNA, right? So this isn't a Chris Angel or David Copperfield, magic trick that I'm doing today, right? We're doing real science. Okay. So what was the purpose of examining it? To prove to you, right, that there's no protein in there, it's just DNA. Step 7. I'm going to give you 10 microliters, so this is a measured amount of the plasmid, into your plus only tube. We're going to mix that up a little bit. Normally we would do that with a loop. Today I borrowed from the biotech lab some micropipetters to make it easier today. What could you add to the DNA negative tube so that you're treating it similarly to the DNA positive? What do you guys think? We're not going to put DNA in there. What could we add liquid that wouldn't affect? Broth, water, sterile broth. At this point we don't, or we could add a little bit more of transformation solution, right? Something that wouldn't cause, right, any discrepancy. This is such a small volume, we don't worry about that volume making a difference in our experiment. So we're not going to add anything to it. So it's a negligible small amount. So, presence of genes that lead to ampicillin resistance and green fluorescence. So before transformation, remember, they're not going to have that enzyme BLA, right? They can't destroy ampicillin. After, they should. Before transformation, do they have the ability to make green fluorescent protein? Before transformation? No. After, should they? Yes. Right? That's the whole purpose of this, is to give them this genetic information. Alright, step 8, you're going to leave the tubes on ice for 10 minutes. Okay? Give them a chance to get acclimated with that DNA floating around in the tube. Step 9, we would normally label your plates. They've already been labeled for you too. And that's because each plate has different media in it, so it's easier for me to label them ahead of time. Keep track of them. You're going to bring your whole ice water bath to the heated water bath I have back there. You'll notice I have it set. It's reading 43, but I bet you it's more closer to 42. I don't think the thermometer's accurate in it, but you'll see I have a thermometer sticking out of it. So that's our warm water bath. So you'll pick up these whole little white racks, they float, you'll plunk it right in the water bath. You will not walk away because you're only leaving it in there for 50 seconds. Right? So somebody in your group needs to time it, and then you're going to take it right directly out of the water bath and put it back on your ice bath. And then you're going to let it sit in the ice for 2 more minutes on your bench top. What's the purpose of transferring these tubes from the ice to the hot water back to the ice? It's what we call heat shock. And as the name implies, this is stressful, right? This is encouraging the bacteria to take up the DNA. So this is almost equivalent to if you've gone on vacation and you jump from the hot tub to the swimming pool to the hot tub. Yeah, it's fun. It's shocking to your system though, right? And it's quite shocking to these bacteria because literally this is temperature equivalent to going from the swimming pool to the hot tub back to the swimming pool. So why only 50 seconds? Because this is about as long as needed for optimum transformation. And it significantly falls off if you walk away. And I've done this in the lab. Right? You don't want to do that. You think, oh, I could go do something. No. You don't have enough time. Stay put. So after those 2 minutes on your bench top, you're going to remove the tray out of the ice and you're just going to put it on your bench. And then we're going to feed our bacteria. So I have little containers that contain sterile broth. And you're going to transfer 250 microliters into each one of your tubes. You're going to feed both your sets of bacteria. Your negative, your control, and your positive test sample. And then, of course, we need to give them time to enjoy the food we just gave them, right? So we're going to give them 10 minutes to do so. Today, again, we're not going to bother with using the crappy plastic pipettes. We're going to use real pipette aids and real pipette tips to do this today. So we'll share, but it doesn't take long. If you don't place the pipette into the liquid in the tubes, then you could use the same pipette. And we're going to do the same thing. We're not going to contaminate. We're just going to squirt it in. So we can use the same pipette tip for both your positive and negative samples. Does that make sense to you guys? Okay. So the pipettes that we would normally use, right, these plastic ones, are not very easy or accurate to use. So we don't even need this picture today, so we're going to skip those. It's a nightmare. So, why are the vials incubated for 10 minutes? What are they doing? We gave them DNA, right? We gave them food. So with that DNA, what can they make? RNA. And then proteins. Which proteins are they going to make? The beta-galactanase enzyme, I hope so. Right? Because if they don't, and we put it on plates containing ampicillin, what's going to happen to those cells? They're going to die. So this is giving them time to make that protein. Are they going to make the GFP? Are they going to make the green fluorescent protein? What do they need to make green fluorescent protein? What has to be present? The sugar, arabinose. Did I say there was any sugar in the media we're feeding them? No. And there isn't. So they won't make green fluorescent protein. They're just going to make beta-galactanase. Then you're going to plate them. We're going to use the special little blue pipetters you guys have on your bench top, on top of your boxes. Those are preset to 50. So if we want to do 100, we have to do it twice. Y'all can handle that, right? 2 squirts onto each plate? I think so. So again, we don't have to worry about that. And since your plus and minus ones are the same, you can use the same pipette tip for both your plus samples and both your negative samples. You'll use the same pipette tip. But you got to switch tips between positive and negative, right? We don't want to cross-contaminate. Because you're actually going to plate the positive sample onto 2 plates. The negative sample onto 2 plates. Right? So you'll do your positive plates, and then you'll do your negative plates. You're going to use a sterile loop just to spread out the liquid that you've pipetted onto your plates. And again, for your plus plates, you can use the same loop. Right? For your negative plates, you've got to use a different loop. Do not tape the plates together as it says in the protocol. Drives me nuts. I've already numbered your plates for you as well. Normally I would have you guys do that, but today I'm doing a little experiment. One of the groups is special. So I want to see if something will come out. So I'll share it with you guys next week if it works. And I don't want to influence you guys in any way, so I'm not letting you know who's the experimental group. So what information is provided by our LB negative plates? So LB is the food we're using. Negative means that we're going to be putting from the negative tube onto that plate. What does that mean? This is going to let us know, right, that the E. coli is still viable after being heat shocked. So basically, you know, we started with good bacteria, we didn't kill them, right? We should get nice, good growth on these plates. Then you have another plate that contains ampicillin. But these are DNA negative. We didn't give them the DNA. So what's going to happen or should happen to these cells? These are our control to show that ampicillin does what to plain old E. coli? It kills it, right? So we should have no growth on these plates. Obviously, transformation can occur only if the PGLO plasmid was introduced into the solution. Which plates are going to be exhibiting transformation? Our DNA plus plates, right? But we have 2 different ones. We have ones that contain ampicillin and arabinose, and one that contains ampicillin and no arabinose. Which plate's going to glow and why? Only the one with their arabinose, because remember, their arabinose is going to bind going to turn on that gene, get the cells to make the green fluorescent protein. The ampicillin plate will still be transformed cells, right? Because they're not going to survive in the presence of ampicillin without that plasmid. So here are our 4 plates, right? We'll see transformation, no transformation on our DNA negative plates. And again, we should get lots of growth. They can grow on the media. We didn't kill them in the experiment, right? Our ampicillin plate should kill our plain old E. coli, our non-transformed. Our ampicillin plate will have only transformed, but remember, no arabinose, no production of the green fluorescent protein, so it's not going to glow when you shine UV light on it. Only our last plate, which contains the arabinose, is actually going to glow. So these show us, right, no green fluorescence because no expression. This plate shows us, right, fluorescence is because of gene expression. So this is hopefully what our plates will look like next time, right? We'll have one plate that glows. So will yours look like this? Hopefully so.